in this module we will see the scoring alignments. So as we have already discussed that the scoring matrices for DNA sequences, and we see that in the scoring matrix of DNA sequences we have used the two terms mu penalization for the mismatches and the sigma penalization for the indels. So this is not the case for the proteins. So for the amino acids, means for the proteins, the situation is more complex as compared to the DNA. In the case of proteins, the common matrices that are available are the pointed accepted mutations, that is abbreviated as PAM, and the block substitution, that is abbreviated as the BLOSSOM. These two matrices usually amounts for the frequency of amino acids X and Y that are present in the evolutionary related sequences. So in the random mutagenesis that is going on in the DNA sequences, some mutations, they are very lethal for the organism, because we know that if there is some mutation in the DNA, that information passes on to the next stage. Means that in the DNA, if there is a mutation, then in the transcription that mutation passes on to the mRNA, and if it is in the third nucleotide of the codons, then that mutation could be very lethal because it changes the amino acid. So this information then passes up to the amino acid, and there is a replacement of amino acid with another amino acid. So in some cases these mutations are very lethal, but in some cases the mutations are not lethal. So when the mutations are lethal, it totally inactivate the proteins. But on the other hand, if the mutations are not very much lethal, then the proteins, they perform their functions very well. So we have seen that in some amino acids, their substitutes are very commonly and found throughout the process of molecular evolution. So as we see here, that some of the amino acids, for example, asparagine, serine, glutamate, and aspartate, they are more mutable, that they have the more ability for mutation, and in the process of molecular evolution these amino acids are replaced by some other amino acids. They have more tendency to be mutated. On the other hand, some amino acids, they are more stable. For example, cysteine and tryptophan, they are less mutable as compared to the other fours. So we can see that if there is a probability of mutation, then serine will mutate and change or replaced by the phenylalanine, and this replacement is 3 times more as compared to tryptophan. Means that tryptophan is converted to also phenylalanine, but this mutation is 3 times less as compared to serine and changes to phenylalanine, because we know that serine belongs to the group of amino acids here that are more mutable, and tryptophan is a member of those amino acids here that are less mutable. So the probability is 3 times more for serine as compared to tryptophan. So knowledge of the types of the changes that are the most common and the most least in the molecular evolution helps the scientists to construct a scoring matrix for the amino acids that can be used to produce the sequence alignment for the amino acids. So as a result, in contrast to DNA sequence alignment, in the case of amino acid sequence alignment, there could be a very few matches. But still, this amino acid sequence alignment could represent an adequate biological sequence alignment. So in the case of entry of the sequence alignment and scoring matrix for the amino acids, that is represented by the delta Ij, and it represents that the I amino acids in the V string is replaced by the J amino acid of the W string. So if we have a large set of alignments of related sequences, then computing delta Ij simply amounts to counting how many times the amino acid I is aligned with the amino acid J. So this amino acid I is from the one string and amino acid J is from the other strings. So in this case, there is a minor complication, is that to build this set of biological adequate alignments, one needs to know the scoring matrix, because we must know the scoring matrix, the mu, the sigma, and what matches one score. So fortunately, in many cases, the alignment of very similar sequences is so obvious that it can be constructed even without a scoring matrix, thus resolving this prerequisite problem of scoring matrix. For example, if we have a proteins that are 90% identical, if we consider this two proteins V and W, and if these are, we can see here that these two proteins, that is almost 90% identical, because in the first amino acid is methionine, in the V and in the W again the amino acid is methionine in the second protein. Second is alanine, here is also alanine, phenylalanine, serine, serine, so it is almost identical. So if these are identical, then even in a naive scoring matrix that we have studied earlier, that gives the premium +1 for the matches, because in match we will give +1 score to this, again match +1, and so on. And plenty is -1 for mismatches and indels. So in this case, we will give the -1 plenty for the indels or the mismatch. But previously we know that there was no such plenty for indels and mismatches. And we can solve this problem by this +1 and -1. So after these obvious alignments are constructed, they can be used to compute a scoring matrix, and that can be used iteratively to construct less obvious alignments. So the simplified description hides the subtle details are important in the construction of scoring matrix. So here we can see here that the serine can be mutated into the phenylalanine, and this is a more common process that diverged 15 million years ago, and this process is going on in the related proteins in mouse and rat. But this probability of conversion of serine to phenylalanine is less as compared to tryptophan that is converted into phenylalanine, and this process was 80 million years ago. But these related proteins that are converting into tryptophan to phenylalanine, they are present in the mouse and the humans. So we can see that and observe that this implies that the best scoring matrix to compare two proteins depending on the similarity of these organisms. So if the organisms are more similar, then they have a less chance to be mutated as compared to organisms that are not similar to each other.